Okay, good morning, everyone. Today, office hours 11 to 12 and 2084 VLSB, and then tomorrow 2 to 3. Also, you can always email me anytime you'd like if you have questions. Those are always useful from my standpoint as they come up, and I'll try to get back to you as quickly as I can. And just see, okay, so this is not Back to the Future here from Monday's lecture. I like to start each lecture with a review of the previous lecture. Also, I guess they had webcast problems, right, for Monday's lecture, as I understand. So between this and what's in the reader, you'll be completely caught up to everything I either said or I should have said on Monday. Okay, so we talked about cell-cell interactions. We talked about 4 kinds of how cells interact in tissues. Gap junctions, which synchronize cells, usually cells in tissues. It's important to point out that free swimming or free cells that are not in tissues do not have gap junctions. 2 examples are spermatozoa and red blood cells. We'll be talking about them over the next week or 2. And they're controlled by a protein called connexin. It's a transmembrane protein. Adherence junctions and desmosomes, both involved in adhesion. Both have cadherin or cadherin-like molecules that we talked about. And they have different kinds of peripheral membrane proteins, catenins and desmoplakin. And then tight junctions are involved in sealing, which is different than adhesion. And the transmembrane proteins are occludin and claudins, and ZO1 is the peripheral membrane protein. I put this up a slightly different way in which we talked about it, so it's a better overview of the issue. But the point is, you need these to synchronize tissue function. It's not fortuitous that all your heart cells beat at the same time, or during labor, all the muscles contract during delivery simultaneously. Okay, they communicate. And they do this through cell-cell interactions and pores. Then we talked about epithelial tissue involved in protection. Tight junctions and adhesion junctions together allow for a selective intercellular permeability, as well as cell polarity, distinguishing different sides of the cell. In much the same way, the bottom part of this classroom is different than the ceiling. Okay, so you have different functionalities that take place. You also have massive secretion and absorption through a lot of epithelial cells. Shape is related to function. So you have squamous shaped cells, which are tile-shaped. And these guys are very leaky. And they want to be leaky. Leaky is good for capillaries, because that's how capillaries can communicate information from different organ systems. And we'll talk a lot about that in the last couple of weeks, actually. You also have stratified squamous, or multiple layers, or skin cells. Skin cells are a little more leaky than, say, other kinds of epithelial cells, not as much as a capillary, because they have a lot of desmosomes that keep them kind of compact together. But the advantage of the stratified part of it is that there's a lot of abrasions. Okay, you fall and you scrape off some skin, your skin grows back in. So you have multiple layers to protect you. The big thing about epithelial tissue is it protects you from extracellular stress of all sorts. Not surprising, as I mentioned, most cancers are epithelial derived, because cancer is a disease of stress and aging. Epithelial cells see the stress directly. Therefore, they're the ones that mostly transform into cancer cells. So the vast majority of cancer cells are, in fact, epithelial. As you might expect, there are defects in gap junctions, adherence junctions, desmosomes, and tight junctions in cancerous cells. That's one of the issues that will fall apart, as in other diseases. Then we talked about connective tissue, which is basically a cell with an extracellular matrix. This extracellular matrix can take on many shapes and forms. So you got bone and is your extracellular matrix from osteoblasts. Fat droplets come from adipocytes. Plasma from blood cells, so your extracellular matrix can be liquid, as we talked about. Fibroblasts, importantly, produce a lot of protein fibers, collagenous, elastic, and reticular fibers. And this holds all your organs together. It's very important part of what these guys do. And then we talked briefly about nervous tissue, that it's multiple cell types. You got neurons and cells that are not neurons. And glial cells is more of a generalized type of cell. And we talked about functionality of the dendrite, the axon, the myelin sheath, what an action potential is, synapse and neurotransmitters. One point I did not mention, neurons clearly have action potentials, but other cell types can also have an action potential or an action potential-like mechanism. And we'll talk about that in the heart cells as it feeds into the pacemaker in the heart, for example. And certain kinds of endocrine cells can have small action potentials. So it's not limited to nervous tissue, although all the neurons will display it more or less. Then we talked about muscle cells, skeletal, smooth, and cardiac. And I think it's pretty clear cut what the differences are. Voluntary, cardiac is your heart. It's contractile and smooth. You don't think about it, but it works. Okay? And then didn't have time, but I just want to briefly talk about the fact that organs have multiple tissues. There's 4 tissue types, but organs, they work together. One example of many examples is the stomach. Here's the lumen in the stomach, and we're going to look at a cross-section of the stomach itself. So here's the lumen. And there's 4 layers. There's a mucosa layer. And if I told you the mucosa layer is involved in massive secretion, gastric juices, so forth, what would be your prediction as to the tissue type and the shape? Any guesses? It's epithelial tissue, right? But what do you think the shape is, given the function that I drew on the board there? Yes? Columnar. Yes, exactly. It's columnar because it's involved in secretion and absorption. In the case of the stomach, it's mostly secretion. When you get to intestinal cells, there's a lot of absorption. So these guys are columnar epithelial cells. Okay? And right behind it, something called submucosa, which is connective tissue and nervous tissue. And you've, of course, got to have a muscle component because you have to move the food through the stomach. So it's muscularis. And what kind of muscle do you think this is? Skeletal, cardiac, or smooth? Smooth, right. Cardiac is obviously the wrong answer here. It's the stomach. Okay? As is skeletal. And then in the bottom end, you got serosa, which is the outside of the stomach, which is a combination of connective and epithelial. This is more for protection and barrier. The point is, typical of all organs, it's multiple tissues. Generally speaking, it's not one type. And they all have to work together, communicate, and do what it takes to keep you going as an organism. So anybody who have eaten breakfast this morning should be very happy about this combination of tissues at the moment. Those of you who are going to eat later will be happy then. Okay? Any questions about this? This is one of multiple examples. In fact, I doubt if you can name an organ that doesn't have at least more than one tissue in it. Okay? So, for the rest of the lecture and through Friday, we're going to discuss the point of view, you are your hormones. Hormones are physiological communicators. They regulate everything from functionality of your cells and tissues to your behavior, so forth. And let me ask you a question. Don't raise your hand, just think. How many of you think you've undergone some sort of hormone therapy? Don't answer that. And also, this is not an eye clicker experience either. Okay? Now, how many of you, when you have a bug bite, go get some anti-itching cream and from the local drugstore or whatever, or the Union here? You can raise your hand on this one. How many of you use bug bite cream, right? Okay, that's a form of hormone therapy. What you're putting on are glucocorticoids, or a form of glucocorticoids that has an anti-inflammatory action to it. Okay? So they're prevalent. Those of you going on to med school and becoming a career as a physician, a lot of what you're going to do is give prescriptions for hormone or hormone-like compounds for lots of stuff. Anti-cancer therapies, a lot of them are very much hormone-related, particularly in breast cancer and prostate cancer. We'll talk a little bit about that perhaps today. There's all sorts of diabetics who take insulin. Insulin's a hormone. Use it to control your physiology. So you are your hormones. Now, you also have the power, unfortunately, of abuse of hormones. For performance-enhancing drugs are really hormones. Okay? And a lot of people, if you take extra androgens, for example, you can get extra muscle mass. Also, human growth hormone allows you to recover from your injuries a little better. But you have the ethical issue of and some of you may be confronted with this, you have a normal-sized person who wants growth hormone right before puberty, so instead of being 5'9", they want to be 6'9". Okay? It's possible to do that physiologically, but it's completely unethical. Okay? So the point is, you can control the physiology and the behavior. And behavior is, from the standpoint of people on performance-enhancing drugs, androgens, have very aggressive behaviors, can have very aggressive behaviors. But there's also physiological downsides. Extremely high risk of cancers. Okay? For men, you can grow breasts. Women can go bald. Okay? There's nothing wrong with being bald, trust me. But maybe not so great on a female. Okay? So the point is there's physiological downsides to all this stuff. So keep in mind that it's a powerful control of your physiology. And this is why it's important for the next 2 lectures to go into this in a little more detail. Okay? Any questions on that? All right, so hormones. What's the overview of hormone action? So we're going to talk mostly molecular mechanism here. The short version of hormones is hormone, and H will be hormone, binds to a protein called a receptor. So you have a binding reaction. You then have a change in the cell, and it's due to interaction with cell components. Steroid receptors interact with DNA and protein. Cell surface receptors we'll talk about. There's changes in phosphorylation events, regulation of secondary messengers, etc. And from this, you get a response. And the selectivity of the hormones. So not all cells respond to all hormones. You have hormones floating all over the place, but not every cell responds to it. And this response is due to the hormone type. So for example, having a lot of glucocorticoids around, as we talked about, has an anti-inflammatory reaction, whereas insulin affects glucose uptake. And those of you who have eaten breakfast have a lot of insulin. Those of you who have not had breakfast yet have a lot of glucagon right now. Very different. One, you're hungry. One, you're well fed. So the hormone type plays a role in the final response. Also, the presence of the receptor. Not all cells have receptors. So an important hormone such as glucocorticoids, virtually all cells have receptors. It's involved in anti-inflammation. It's a systemic approach to the stress. But not all cells have androgen receptors or estrogen receptors. Not all cells have growth hormone receptors, for example. So the presence is very important. You also have hormone receptor binding affinity. How one responds to these hormones can be different from person to person. It's because the affinities can be slightly different. There's also lots of mutations out there where the affinity of the receptor for the hormone is altered, and therefore you have a possibility of a genetic defect. And we'll talk a little bit about that if we get to it today, certainly on Friday, of AIS, which is androgen insensitivity syndrome. Part of it is due to producing receptors that don't recognize the hormone very well at all, and has a very dramatic phenotype. And finally, another part of this is how the receptor interacts with the cell components. So you can have a normal hormone, a receptor, but if it doesn't interact with the cell components properly, you can have problems. So a lot of type 2 diabetes, adult onset diabetes, you have a normal hormone, a normal receptor, but it's the downstream cell components that are messed up a little bit. And it takes a while to manifest that, and then that's why it's adult onset in the diabetes. So it's not necessarily the receptor or the hormone is the problem in a lot of cases. It's what's downstream. It's what's in between the receptor and the response. And that's the harder part to treat, but it's a very common kind of defect in these physiological disorders that relate to hormones. Okay? Questions on that? Questions on that overview? Okay, so where do the hormones come from? And our point of view of hormone action is going to be the target tissue, but I think it's important to see what's going on and where they come from. The classical pathway is called the endocrine pathway, and is where the hormone is produced by endocrine cells or endocrine glands. They synthesize the hormone, and it travels through the blood. Reaches a target tissue, and the target tissue is defined as having the receptor. No receptor, not a target tissue. Has the receptor, is a target tissue. Even if it's not functional, it's still considered a target, in my opinion. Now, in the endocrine cells, let me just do this here. I can do this down here. On the endocrine cells, there's two ways in which the cells can sit in these endocrine glands. And that is, they can be clustered, forming an organ, an obvious organ or an obvious gland. An example is the anterior pituitary, which we'll talk about starting today a little bit. You can also have dispersed endocrine cells. An example would be the endocrine cells in the testes or the ovary, for example. Another one that we'll also get to on Friday, testes and ovaries we'll do next week, with human reproduction. So we can have dispersed, and these would be the alpha and the beta islet cells of the pancreas. And these are the ones that produce insulin and glucagon. So those of you who had breakfast this morning, your beta cells are secreting insulin. Depends how early you had breakfast. If you had breakfast like when I had at about 4 in the morning, I'm probably getting closer to my glucagon than my insulin right now. And alpha cells are producing the glucagon, for those of you who did not eat breakfast. Okay? The point is they're dispersed throughout the pancreas. They're not an identifiable organ or a gland. Okay? Now, these cells to this target tissue can come from different places. This is the classical pathway produced by an endocrine organ. Hormone gets dispersed in the blood, binds to receptor, you induce a response. You can also have a neighboring cell. So here's the cell with the receptor, and here's a neighboring cell that produces the hormone. This hormone could be identical to the one that comes from an endocrine gland, and the receptor can be identical in the target cell. And one way to think about this is in a cell tissue, you have all these cells lined up next to each other. One cell might have one of the key receptors, and the other cells nearby can produce the hormone inside the tissue. This is an important issue because a lot of cancer cells, when they first transform, they're sitting in a tissue. And what they're doing is, a lot of times, is at least initially, they respond to the hormones that are produced from the normal cells to keep the cancer cell, for example, to grow in an uncontrolled way. So this kind of pathway called a paracrine pathway, that does not go through the blood, is very important. Everything from tumorigenesis to keeping normal tissues in its correct architecture and function. Another kind of paracrine system is the synapse, is the neurotransmitters at the synapse. When the neurotransmitters come across. Some people give it a different designation. I just consider the synapse a unique kind of paracrine hormone system because it's not going through the bloodstream. So a lot of your hormones do not have to go through the blood. At least I don't consider it at all. And finally, you have something where a cell, pretty typical of cancer cells actually, the cell has a receptor, makes its own hormone, and the hormone comes back to the same cell. So the same cell is making the hormone as produces the receptor. So it's auto-regulating. And when cancer cells, particularly later stage cancer cells, they produce a lot of their own growth factors and respond to their own growth factors. And this is called autocrine. Okay? In all cases that I have on the board here, the receptor can be absolutely identical. whether it's an autocrine, a paracrine, or an endocrine system. Hormones could be quite similar, if not identical. So it's not only coming through your blood, although that's the classical definition. It's clearly recognized that paracrine system and autocrine systems are quite important in the normal tissue function and in disease states such as cancer. Very important part of this. Any questions on this point of view? Yes?
These are all endocrine the classical endocrine pathways, or that was the first one?
Yeah, the classic endocrine pathway is the hormones traveling through the blood. What has been recognized in the last, I don't know, 20 years, 25 years or so, doesn't seem that long to me, but I know from your standpoint it's a pretty long time ago. Hormones can come from cells next to each other or from the same cells. A very important part of regulation. Okay? It's all happening together. Okay? It's not just either, it's not an either or. It's all kind of happening together. But this is the classical pathway here. And that's the name, endocrinology. Comes from the classical pathway. If they had discovered this first, probably the whole field would be called paracrinology or something like that. Okay? Yes?
You said that the receptors may be the same for all of these pathways?
Yes, the receptors can be the same for all the pathways.
And did you say the hormones can or cannot be the same?
The hormones sometimes are, sometimes are not. As an example of the complexity of the answer to your question, it's a very good question. A lot of times the hormone that's coming through, like, say in this pathway, might be epidermal growth factor. Okay? But in a cancer, and some, maybe even the paracrine pathway could be epidermal growth factor, binds to an epidermal growth factor receptor. But a lot of times in the autocrine pathway, instead of epidermal growth factor, a very related hormone called transforming growth factor alpha has identical functionality as epidermal growth factor, binds to the same receptor, but it's a slightly different hormone. There are other cases where it's exactly the same hormone. Our point of view is it's the target cell. The cell with the receptors is what gives you the information and the response. Okay, so if you don't have a receptor, it doesn't matter whether the hormone's around or not. You're not going to respond. Herein lies a lot of genetic disorders. Okay? So, what does this all mean? This is the backdrop to the issue of homeostasis. It's a term I'm sure you've heard. Homeostasis is your steady state. All of you have a homeostatic state, but all of it is slightly different from one to another. What this really is, to maintain homeostasis or maintain your steady state, you have dynamic changes, dynamic responses, probably a better term, to various stimuli. And a lot of these stimuli tend to be hormones. Not all of them, but a lot of them are hormones. And you do this through feedback mechanisms. And we're going to talk briefly about this because you're going to see this time and time again. You cannot understand human reproduction, at least the hormonal part of it, without understanding feedback mechanisms. Okay? So one type of feedback is called negative feedback. The strict definition, I'll put this board down in a second. The strict definition is a change in a variable reverses the change. Okay? What's very important to remember here with negative feedback, the object of negative feedback is to maintain a constant level. Although it seems like an inconsistent term, negative feedback, what it means physiologically is maintaining a constant level. And that's very important to see this because of the organ systems that are involved. So let me give you the more generic example here. This constant level means physiologically it's never a constant level. It's maintaining the level of a molecule or whatever within a window of acceptability. So constant does not mean a straight line. It means maintaining some molecule within a window of acceptability. Okay? It's very important physiologically. So for example, here's the window of acceptability. And if you go outside this window of acceptability, you might have a disease state or some sort of physiological disorder. That's why when you go to the doctors and you get a blood test, and they say, oh, you have the level of X, Y, or Z is within the correct range. This is what they're referring to. So in terms of negative feedback, what this means is, say you have this molecule X, and say X is low. Say it's down here. Your system then makes more X. And if it gets to the upper reaches of this window of acceptability, you now decrease the synthesis of X, and you bring it down. And then when it gets too low again, you increase X. And then you decrease it again. It keeps going on and on. And this is due to a negative feedback. So if you have too little of it, the negative feedback says make more. If you have too much of it, the negative feedback says let's reverse it, right? You're reversing the change. Reverse it and you come back down. Too low again, reverse it, you come back up. Okay? So negative feedback physiologically means maintaining the level of a molecule with an acceptable window. You will see multiple examples of this, including one today. Yes, question? Question is, how do we go out of the window? Well, we don't want to go out the window, or out of this window. Maybe other windows are fine, but not this window, okay? When we go out of this window, we change the physiology. You'll see a very clear example of that next week in female reproductive system on the menstrual cycle. That just a little bit of an advertisement, I guess, for next week. You stay in this window at a certain level of estrogen. And as soon as your estrogen levels go too high, you're out of this window, and you immediately kick on ovulation, for example, which is different than what's going on during the normal part of the window. Okay? When you're eating, insulin levels come up, they come back down. They come up, they come back down, depending if you're eating. What if you're eating, and instead of going back up, they go down? You can't bring in glucose, you're going to be diabetic. So you want to be inside this window. Now, this window means different things for different people. The concentrations of the window are different. I could say on an average of 1 million people, I can get a certain window of a certain molecule. But for any individual, like your two neighbors sitting next to you, your window of acceptability for a given pathway might be quite different. But it's normal for you. Very important to see that. As a doctor, you guys will become physicians, you'll see this time and time again. Question?
What actually detects this negative feedback?
What detects the feedback? We will talk about that in a second. What detects it is different for different systems. Human reproduction cannot even be thought about in terms of understanding it without understanding that exact point, what detects it, and so forth. As I'm clearly leading to, what detects it are the receptors in the organs that react to the X, and X being the hormone. Okay? So this is all coming around here to what we've been talking about. So for example, a negative feedback system we will talk about in multiple situations is the hypothalamus anterior pituitary axis. So you have the hypothalamus, and this produces something that's historically called releasing factors. But what are these releasing factors? They're hormones. Just because the old name calls them releasing factor doesn't mean they're not really hormones. These will bind to receptors sitting in the anterior pituitary. Causing the anterior pituitary to then produce more hormones, which hits various organs or other endocrine glands. Call these either glands or organs, doesn't really matter, gland, endocrine gland, to produce yet another hormone. So we'll call this first hormone hormone, second hormone H2, And the hormone that this final gland produces is H3. And H3 will have a bunch of effects on your system. In case of male reproduction, it's androgens. Okay? In case of females, a lot of times it's estrogen or estrogen and progesterone. What this hormone will do now is you're making this hormone. You got to bring it back down, right? Negative feedback says you got to start bringing this back down to keep it within your acceptable window. So this hormone then feeds back and prevents the anterior pituitary from making hormone 2 and feeds back to the hypothalamus and prevents the hypothalamus from making what's called hormone 1 in this discussion. So if hormone 3, which is affecting your system, is too low, the negative feedback is not available. Hypothalamus makes more H1, anterior pituitary makes more H2, you make more of H3, and you have your systemic effect, wherever that is. But if this gets too high, you don't want to go above your acceptable ceiling in your window. When this gets too high, you feed back, shut down hormone 2, shut down hormone 1. Does everybody see that? So it's the hormone and receptor systems that keep it within this acceptable window of negative feedback. Questions? So I'm going to give you a specific example here in a second. Okay, so that example I want to give you is long-term stress. The hypothalamus in long-term stress, and this could be, for example, you have a flu or a cold, could be a bacterial infection, could be anything. It could be over-worrying about studying can affect this to some extent, okay? Lots of stuff. The releasing factor that gets produced is called CRF, corticotropin releasing factor. This is a hormone, even though it's called historically a factor. Binds the receptor in the anterior pituitary, releases ACTH, adrenocorticotropin hormone. Okay? Just ACTH is fine. By the way, in any exam I'll give you, it's not going to be a spelling test. Whether it's corticotropin or corticotrophic, releasing factor or releasing substance, nothing like that. Okay? CRF gets produced to the anterior pituitary, binds the ACTH. The gland is the adrenal gland, right above your kidneys. In fact, the adrenal cortex, which is the outside, let me just put this up here. So in your adrenal gland, the outside is the cortex, the outside part of it, and the inside is the medulla. And the reason I bring this up is we're going to talk about short-term stress at some point, which affects the medulla. The adrenal cortex, when it sees ACTH, converts cholesterol through a variety of steps into your hormone 3, which is cortisol, which is a glucocorticoid. This, by the way, is a synthetic version of this, is the component of that anti-itching cream I was talking about earlier in the lecture. Cortisol has systemic effects. Brings in glucose, anti-inflammatory effects, has immune system effects, etc. But when the glucocorticoid levels get too high, feeds back and shuts down ACTH production and shuts down CRF production. So you keep glucocorticoids within your acceptable window. If you have too much glucocorticoids, there's diseases, syndromes out there that involve way too much glucocorticoids and therefore too much of its systemic effects, therefore not good for you. Okay, questions on this? Okay, good. Now, unlike negative feedback, you also, was there a question? Oh. Unlike negative feedback, you also have positive feedback. No surprise. And here, the change in the variable, instead of reversing the change, amplifies the change. So when would you ever need positive feedback? Well, one very clear example of positive feedback is during labor. So here's the baby coming down the birth canal. And we'll give him or her a little smile. I have four kids, and I was at every single birth. Well, actually, it was 3 births because I have a set of twins. And trust me, they are not smiling on the way out here. So they're getting ready for this, and you have pressure of the head. And the pressure causes the posterior pituitary to secrete oxytocin, which gives you contractions, which causes more pressure, which causes more oxytocin, which causes more contractions, which causes more pressure, more oxytocin, and more contractions. And it keeps going in a positive feedback. It amplifies. It gets more difficult, stronger and stronger, and then the baby's born, it all dissipates. And that's a good way of thinking about positive feedback, because that's why the contractions get stronger. Talk to your moms about that. Okay? The point is, it's all because of positive feedback. There's more to it than oxytocin, which we'll talk about next week. But it is the primary hormone involved. There's another form of positive feedback we'll talk about in the menstrual cycle. The last part of the menstrual cycle, or the middle part, is a positive feedback involving estrogen, where you shift from a negative feedback to a positive feedback. And that happens during the menstrual cycle. And we'll talk about that next week. The point is, lots of examples. Okay, questions. Difference between positive and negative feedback. You've got to imprint this in your psyche because it explains a lot in terms of how different systems work physiologically. And those of you going into medicine will see this time and time again. Question? In this case, the variable dissipates. In this case, the baby's born. In the case of the female reproductive cycle, ovulation. So there's a big event will take place. Okay, very good question. As you can imagine from this, if you want to induce labor, you give them an oxytocin or a version of oxytocin as one approach. But we'll talk about that next week. But you can almost could have predicted that from this discussion. Okay? Now, you also have sometimes no feedback. You're just responding to the environment. And that is, one example of that is your short-term stress, your rapid stress response. Do any of you, before you go in for an exam, right before it's handed out to you and you're sitting there, you're a little nervous and so forth, do you feel your heart beating a little faster sometimes and everything? Yeah? Okay? That's a rapid short-term stress response. It's not really involving a feedback, but it is a response to an external stimuli. So here the hypothalamus, through nerve connections to the spinal cord, and let me put this down here, and from the spinal cord to the medulla of the adrenal gland. Okay? And what it causes the medulla to do, and I'll put this here, is you convert tyrosine, which is an amino acid, to epinephrine. And epinephrine then goes throughout the system and has a lot of effects, quickly secreted. And it has a rate of increase of your heartbeat because it targets the pacemaker in your heart, for example. High metabolic rate. It gives you a state of alertness. That's the part I always liked about it. You get acuity and alertness, at least momentarily. It's always nice momentarily to have that. Okay? So it's a very rapid, quick response. It's not necessarily a feedback as it is to respond to the environment. Okay? So there's no particular feedback involved there. Any questions on that? Okay. So in terms of hormones, in terms of thoughts on it, A lot of the hormones will bind to receptors I mentioned and give you a response. These are called agonists. The majority, all the hormones that we talk about, you have a few examples, but most of the hormones in your body are agonists. Insulin, estrogen, glucocorticoids, androgens, they're all agonists. A lot of times medically you want to counteract what's going on with the hormones. And what you have also is, most of these are synthetic, but not always, antagonists. So an agonist binds the receptor and induces a response. An antagonist, there's different kinds. We'll talk about really one kind here, called an anti-hormone, binds the receptor but gives you no response. And in fact, prevents a hormone agonist from working. And medically, these are very, very important. A lot of your anti-cancer drugs, or some of them, are based on being antagonists for a receptor that's important for the growth and function of a cancer cell, as an example. Many examples of this. So for example, in breast cancer, I'll put this up here. So in the case of early stage breast cancer, estrogen, which is a steroid, E for estrogen, binds to a receptor and causes a breast cancer cell to grow. To actually proliferate is the correct term here. Usually earlier stage breast cancers. And this was recognized early on. And have any of you guys ever heard of tamoxifen or raloxifene? Those kind of molecules. These are anti-estrogens. So tamoxifen, they give, which it is really is an anti-estrogen. Oh, technically, it's not as considered as strong of an anti-estrogen as it used to be. Will bind to the estrogen receptor, prevent estrogen from binding, and shut down the growth of these early stage breast cancer cells. And many, many women have been given tamoxifen to stop the growth of estrogen-responsive breast cancers. It works because it's an antagonist. As an aside, tamoxifen binding to the receptor causes a different three-dimensional conformation of the receptor than estrogen, binding to the same receptor. That's why it works. You don't need to know that, but those of you getting into this professionally, that'll become a big issue. Try to develop better anti-estrogens for this treatment. Okay? Another antagonist that's known out there is RU486. Have you guys heard of that? It's the abortion pill. Progesterone is important for implantation of the placenta in the uterine lining. And if you give an anti-progesterone, RU486, you can disrupt the implantation. So here's a multi-billion-dollar industry based on antagonist function versus an agonist. So a very important part of this whole thinking about things medically. Okay, you'll see this time and time again. Okay, any questions? Okay, so now we're going to go slightly deeper. So we've been mentioning some steroids. We're going to go a little deeper into steroid hormones. And the receptor action. Okay, what are some of your favorite steroids? If I said to you steroids, what do you throw back at me? Estrogen, what else? Testosterone, okay, you got estrogen, which is considered the female sex hormone, but males do make it. You have testosterone or androgens, which is the more generic term. It's considered the male hormone. However, every female that makes every molecule of estrogen makes a molecule of testosterone because testosterone is the precursor in females to estrogens. So to say that androgens is strictly a male hormone and estrogen is strictly a female hormone is technically incorrect. But it is some of our favorite steroids, nonetheless. Can you name any others that we even mentioned today? So if I said to you long-term stress, what would you say? Glucocorticoids, right? Involved in long-term stress response, immune system. You also have progestins, or progesterone, which is actually the pregnancy hormone, but it's also the precursor for a lot of the other steroids. And you also have mineralocorticoids. At some time or another in the next week or so, we'll mention all these hormones. Certainly next week, estrogens and androgens will be critical for human reproduction. Same with progesterone for pregnancy. Mineralocorticoids are involved in blood pressure, blood volume regulation, salt balance. Glucocorticoids involved in stress. Now think about the biological range of physiological responses, right? Female reproduction, male reproduction, pregnancy hormone, immune response, sodium retention, et cetera, right? That's a broad range of physiology. What's interesting is all these guys work through a very similar mechanism because these steroid receptors, call this SR, for steroid receptors, all are transcription factors. So the receptors themselves are transcription factors. If I say to you transcription factor, what's the first thing you say back to me? Or hopefully one of the first things you say back to me. Huh? Well, before mRNA, promoter DNA binding, right? You guys had that? Probably way too detailed previously in this class, okay? But these guys are all transcription factors. So here the hormone comes in and basically turns on a transcription factor that we historically call a receptor. And this is important because there are a lot of important value, physiological value and disease states involved in this. And how these work in a general way is these steroids, here's a plasma membrane, come from the outside. If you look at a structure of steroids, and the reader I threw some structures in there, and you do not need to know the structures before anybody asks that. If you look at the structure, however, it's a multi-ring hydrophobic molecule. So it's a little greasy molecule. It loves to get through the plasma membrane, or at least get into the membrane, where it binds to the receptor. This is my, it ages me, but this is my Pac-Man model for the receptors. I actually, in the 70s, played Pac-Man, put quarters in there back then, stuff like that. Got good at it for a while. But that's a piece of trivia you don't need to know either. So this receptor then comes down. Upon hormone binding, there's a change. And you don't need to know what this change is. There's changes in the protein complex, changes in protein interactions. There's a change takes place, and oh, there's the blackboard. A change takes place, and this receptor can come in, sit down on the promoter of a gene, a lot of times it can be a homodimer. You can have an SR homodimer sits down on the DNA. We're not going to worry about that too much, the distinction. But it will sit next to a transcription factor. TF is a transcription factor. This is a generic term. And you should have, as was implied by the answer to my question earlier, these transcription factors bind to DNA also. So what happens is, on the promoter of this gene, you form a protein complex. I'm drawing this as a two-protein complex. The reality is, you don't need to know this, but except for later on, the reality is it's about 50 proteins or so will form a complex. It's very complicated in a lot of ways. The binding sites, the DNA sites, are specific sequences. And we'll end with this term of what is the nature of the specific sequences. For these receptors, they're given a generic term of HREs. And what's an HRE? This is, again, a generic term here. hormone response element. When you see the word element, it generally means some sort of specific DNA sequence. If I say to you it was the glucocorticoid receptor, what do you think they call it? This is where we'll end. They call it a glucocorticoid response element. If it's the estrogen receptor, what do you think they call it? Estrogen response element. Okay, keep that in mind, and we'll pick this up on Friday. And again, office hours 11 to 12.